Hello students and welcome to the program of Bachelor of Science 3rd year, subject Botany, semester 5, paper code BOC 107 and paper title Microbiology and Plant Pathology. Title of the unit, Unit 2 Methods in Microbiology, module name Types of Culture Media and module number 06. In this module, I'll be defining the culture media, talking about the characteristics of media and explaining the classification of media based on composition, consistency and function. At the end of this e-module, the student defines the different terms, lists different types of media, classifies types of media and differentiates between different types of media. What is culture media? It is a mixture of chemicals that support the growth of microorganisms. These microbes use the nutrients present in the media as their food required for their growth in vitro. Now coming to the characteristic of media. The media should neither be acidic nor alkaline or basic in nature. It should contain all the nutrients required for microbial growth. And most importantly, the media should be sterile, that is, it should be microbe-free before use. Now coming to the classification of media. Media is classified broadly on the basis of chemical composition, consistency of media and function. Now, classification of culture media on the basis of chemical composition. We classify it into two types, synthetic or chemically defined media and complex or chemically undefined media. Synthetic media is a media in which the exact chemical composition is known. That is, the pure chemicals with the known concentration are taken to prepare the media. Example, Zipax media. Complex or the chemically undefined media is a media in which the exact chemical composition is not known. Along with the pure chemicals in this media, the plant products or the animal products are added. Example, the potato dextrose agar, or it is also called as PDA. The synthetic or the chemically defined media, it is generally for the bacteria, and the chemically undefined media, that is the PDA, is for the fungal culture. Now coming to the classification of media on the basis of consistency. It is classified into solid media, liquid media and semi-solid media. Solid media is a media in which the solidifying agent like agar is added. When this media is cooled down after sterilization, it solidifies and we get the solid media. Liquid media is also called as broth. In this media, the solidifying agent like agar is absent. As such, when the media is cooled down after sterilization, it remains in a liquid state. Semi-solid media is also called as a floppy media. In this media, the half-strength agar or the required quantity of the agar is always added in half quantity. As such, when the media cools down, neither it remains in a solid state nor it remains in a liquid state, but it is in between, that is semi-solid. Now the classification of media on the basis of function. They are supportive media, enrichment media, selective media and differential media. Now what is supportive media? In this media, along with the other components, additional ingredients like blood is added. And with this additional ingredient, the microbes will grow very fast. That is, this media will always support the growth of fastidious bacteria, means the bacteria that require the specific requirements in the media. Example, the blood agar. Enrichment media. In this media, number of growth factors are added, and these growth factors will always enhance the growth of the microorganism. Not only that, but will be able to recover the desired microorganism. Again, the example is blood agar or the chocolate agar. Selective media. In this media, the special compositions like chemicals or dyes are added. Because of the presence of chemicals or the dyes, it always promotes the growth of one type of microorganism and inhibits the growth of other type of microorganism. The very best example is EMB agar, or it is also called as eosine methylene blue agar. In the presence of this media, only the gram-negative bacteria will show the growth, whereas the gram-negative bacteria will always be inhibited. Now, example of the gram-negative bacteria which shows growth on this media is the E. coli. So, whenever the E. coli grows on this media, because of the formation of the complex of eosine methylene blue, the colony color will be like it gives the metallic sheen. That is, E. coli colony will be showing the metallic sheen. Differential media. In this media, one microorganism can be differentiated from the other type of microorganism present on the same media on the basis of their biological characteristics. Example, McConkey agar. Now on this agar, the E. coli and the Salmonella typhi, they grow. Now when both these type of microorganisms are growing, we can very well differentiate these microorganisms on the media, and we can differentiate on the basis of their colony color. E. coli will appear brick red in color due to the fermentation of the lactose present in the media, whereas Salmonella will be pale yellow or white in color due to the non-fermentation of lactose. Thus, due to the presence of the different color colonies, we can very well differentiate the two organisms on the same media. These are the few references which I had referred while preparing this e-module. Thank you, dear students.